I welcome all of you to this course unit, which is called IPS 1209, and the name of the course is Diversity and Classification of Organisms, or we can call it Diversity and Classification of Living Things. Like I've already written here, this is a branch of biology which we call taxonomy. Taxonomy is a branch of biology which deals with the science of classification. It is the science of classification of living organisms. That is taxonomy. It is a full branch of biology. And the father of classification was a botanist known as Carolus, like this, Carolus Linnaeus. Carolus Linnaeus is the father of taxonomy, the father of classification. And we can abbreviate that name as simply Carl Linnaeus. So most of the principles of classification which we are going to study were founded by this botanist, Carl Linnaeus, in the 18th century. A century is always 100 years. So it was about 17 in the 17 something like 1776, 1780, 1760. That is what you call the 18th century. So Carl Linnaeus introduced the science of classifying living things on the basis of common features, on the basis of their similarities, on the basis of what they share in common, so that organisms that show resemblances, organisms which show similarities, are placed in the same grouping. They are placed in the same taxonomic grouping. So I would like us to explore or to review definitions of three terminologies. The first terminology is taxonomy, the second one is systematics, and then the third one is classification. So to begin with, I've already defined the taxonomy. It is the science of classification. It is the science of classification. So, a biologist who deals with classifying living organisms, classifying plants, classifying animals that are diverse, but placing them into recognizable groups where the members in that group share similarities. They have some features in common. That scientist is called a taxonomist. So the taxonomists could be for plants. Therefore, we can have plant taxonomy or plant taxonomists. They only deal with classifying plants of various forms. Then we can have also animal taxonomy. We can also have taxonomists of microorganisms like viruses. We can have taxonomists who classify bacteria and taxonomists who classify fungi. So we can have taxonomists of different groupings of living organisms. But the science of classification is what we call taxonomy. The science of classification is called taxonomy, and we shall look at the basic principles of classification, the basic principles of taxonomy. Now, as I was explaining the term taxonomy, I talked about what I called grouping. Grouping. Now, the grouping of organisms, the placing of organisms into certain convenient groups, into groups, placing organisms into groups, is a branch of taxonomy which we call systematics. 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 So systematics is a branch of taxonomy. And what does it deal with? Placing organisms. Placing organisms into groups. Into groups. So we can have, for example, that every organism which is autotrophic, every organism which can make its own food, maybe by photosynthesis or by chemosynthesis, but it has that ability of building up organic compounds from simple inorganic materials obtained from the environment, like those obtained from soil and air. The simple inorganic materials, it builds them up into complex organic compounds, such as the carbohydrates, such as the proteins. Now, that organism is called a plant. That organism is called a plant. So we will place every organism that has that ability, regardless of its complexity. Regardless of its complexity. It may be a simple life form. It may be a simple form of life, such as the algae, which here in Uganda we call, like the green algae, we call it enkonge, enkonge. It could be a water weed, like the water hyacinth. But provided that plant has mechanisms, photosynthetic mechanisms, photosynthetic machinery, it will be placed in the same order. Sorry, in the same group. It will be placed in the same group like a tree, which is a complex or a higher plant form, a higher form of life. So when we place organisms that share certain similarities, that have common features in the same grouping, then that one is called systematics. And it is a branch of taxonomy. It's a branch of taxonomy. The second branch of taxonomy is known as classification. Classification. Now, this term classification simply means placing organisms, placing organisms or sorting out organisms into suitable groups. Sorting out organisms into suitable groups on the basis of their shared characteristics, on the basis of shared features, or on the basis of their resemblances. So we will place all organisms which are heterotrophic, that is, they are unable to make their own food, but they obtain their food, they obtain their nutrients from other sources. We may place all of them in the group of animals. We may place them under a certain grouping of animals. Then I've said those which are autotrophic could be placed under a grouping of plants. Then we could have maybe what we call domains. We can have what we call domains. We can place all organisms with prokaryotic cells, cells in which the genetic material is not found in the true nucleus, cells in which the genetic material is found somewhere in the cytoplasm of that cell, in an area called the nucleoplasm. But there is no membrane-bound nucleus. There is no nucleus bounded by a nuclear membrane, which nucleus contains the genetic material, DNA. So organisms which have that true nucleus, where the genetic material is enclosed within a membrane structure, we call those eukaryotes. Eukaryotes. They have a true nucleus. They have a true nucleus. And what we are calling a true nucleus is one where the genetic material, the hereditary material, DNA, is enclosed by a nuclear envelope or a nuclear membrane. We call those cells eukaryotic cells. And you will find that we have a grouping, what we call a domain, which we call eukarya or eukaryota. Eukaryota. EU. That word EU means true. Then karyota comes from the word karyon, meaning nucleus. So these ones have a true nucleus. And you'll find all organisms whose cells are eukaryotic, whose cells have a true nucleus. And those cells also have what we call membrane-bound organelles. They have organelles, such as the chloroplast, the mitochondria, the ribosomes, the endoplasmic reticulum, the Golgi complex or Golgi apparatus. Those are called membrane-bound organelles within the cytoplasm of the cell. So such organisms with eukaryotic cells, they form their own domain of life. The domain of life, which we are going to see, we can either call it eukaryota or eukarya. Some books, you'll find them using that word, eukarya. Eukarya being made up of this domain of life being made up of the fungi. The fungi are part of eukaryota or eukarya. We have what you call the protists, another kingdom of grouping. We call it protista or the protists. Have you ever heard about amoeba? What we call the single-celled organisms, such as amoeba, paramecium, which are mainly aquatic in damp environments. But the whole body is made up of one cell. Have you ever heard about Euglena or the Euglenoids? All of those are under this kingdom or grouping, which we call protists. And we call them protozoans. So, what do they share in common? Eukaryotic cell. They all have eukaryotic cell. One with a true nucleus, that is one in which the hereditary material, the genetic material, is enclosed in a nucleus, which is covered by a nuclear envelope or a nuclear membrane. Another group, the plants. So you will find that all plant cells have got a true nucleus, which is enclosed, a true nucleus where the genetic material is enclosed by a nuclear envelope, And the other group is Animalia, kingdom Animalia, and these are the animals. So you see, we have placed all these four groups in a large unit of classification. Actually, the largest unit of classification in modern taxonomy, in modern biology, is known as a domain, the largest unit of classification in present day taxonomy is known as a domain. You will find already books talking about a kingdom being the largest unit of classification. But new developments in biology, new developments in taxonomy, in the science of classification, now we talk about a domain as the largest unit of classifying living organisms. If that is the first domain of life, we have another domain of life which we call eubacteria. We call it eubacteria. Another domain is called eubacteria. So, as we are doing this, as we are looking for resemblances among larger groups of organisms and placing them, for example, in the same domain, we are classifying these organisms. So, another domain is called eubacteria. I said the word EU, U means true. So these are the true bacteria. They are the true bacteria which we know mainly inhabiting, they are unicellular. These true bacteria, the true bacteria we know, they are unicellular. In other words, the entire body is made up of one cell. But we also know that some of them are free living. They are free living bacteria, abundant in soil, abundant in air, abundant in water, in most environments. They are there. They are there. It is these bacteria which cause disease in plants and animals. You'll find them causing disease in plants and animals. Other bacteria, you'll find them living symbiotically with the animals, symbiotically in the body of an animal. In other words, they benefit the animal. They benefit the animal. Now, this, for example, include what you call the gram-positive bacteria and what you call the gram-negative bacteria. They are called true bacteria, eubacteria. That is another domain of life. Remember, I've talked about 3 domains of life. And I have begun this conversation of classifying at the largest unit of classifying living organisms, which we call a domain. The other domain, which we call archaea. We can call them archaea or archibacteria. These are not the true bacteria. This is another domain of life. Where you'll find those bacteria like what we call the hyperthermophiles. Thermophiles. Let me write it for you here. Hyperthermophiles. This is a group of archibacteria that live in extremely high temperature environments, environments of extremely high temperature. So we find the organisms in this domain of archaea or archaebacteria being those extraordinary special group of bacteria that can inhabit extremely acidic environments or extremely saline environments, salty environments, or those which can inhabit, those which can occupy extremely uh, hot environments, extremely hot environments. So I've just given here one example which we call hyperthermophile, and others, another one could be like hyper halophile, those which live in marine, extremely marine or salty environments or saline environments. So mainly these archaebacteria, they are not very common in our ordinary tropical environments. No, you'll find them in some extreme environmental conditions. But taxonomists have decided to group living organisms under those three domains of life. The first domain being eukaryota or eukarya. The second domain being eubacteria. I would like to give you another term which you will find very common in this domain. In this domain of eubacteria, you'll find a common term which we call prokaryota. Prokaryota. So whenever they talk about prokaryotes, or prokaryotic cells, these are mainly the bacterial cells. The bacterial cells. Now, that word pro means before. It means before. Karyota comes from the word karyon. It comes from the word karyon. And I said karyon means true nucleus. True nucleus. So when you say prokaryota, these are believed, the prokaryotic organisms or the prokaryotes or the eubacteria are believed to have evolved way back many millions of years ago, before the eukaryotes, In other words, these prokaryotes or the eubacteria evolved at a time when a nucleus, a true nucleus, had not yet developed within cells of organisms. Instead, the genetic material, the hereditary material, was confined, located somewhere in the cytoplasm, in an area which I've called a nucleoplasm. So you will find these ones having a nucleoplasm or something like a nucleoid. That particular location of genetic material, and which genetic material is a bit circular. It is a bit circular, the genetic material DNA. What about the eukaryotes or the eukaryotic cells? You will find them having the genetic material DNA located in a specific region known as the nucleus. And the genetic material here is known as a double strand or a double helix. It is a double helix in eukaryotic cells. What about here? Here it is circular and single-stranded DNA. So when we are looking at the structure of the DNA, it is simple in the prokaryotic cells, because even these forms of life, the bacteria or the prokaryotes, are the smallest. They are the smallest cellular organisms known on Earth, known to exist on Earth. They are simple forms of life. The other eukaryotes, as I've already indicated to you, including the plants with plant cells, the animals with animal cells, the fungi with the fungal cells, the protists or protozoans with the single cells, all of those ones, in their cells there is a true nucleus. And remember, the nucleus is the control center of all the activities of the cell, while the cytoplasm is the site of the cell's activities. The nucleus is the controller. Within the nucleus, we find hereditary material, genetic material. We find that molecule called DNA, whose instructions dictate or determine the type of protein a cell will make, the type of enzyme a cell will make, the process of cell division. So instructions controlling all the cell's activities originate from DNA in the nucleus within eukaryotes. That is why this grouping, the eukaryotes, they are able to assume or to develop into higher forms of life. They are able to assume large size and complex metabolic processes. So mainly the main domains of life, which we know are these two: the eukaryota or eukarya, and the prokaryota or the eubacteria. Because I've said this third domain of life, they are mainly found in extreme environments of high salt concentration, high temperatures, high acidity. So they may not be common within the tropical and even the marine or freshwater environments. So when a taxonomist does that, when a taxonomist begins sorting out organisms on the basis of their resemblances, on the basis of shared features, then we say that taxonomist is classifying organisms. So classification is simply sorting out organisms into recognizable groups, placing organisms into certain groups on the basis of their shared features or on the basis of their similarities. That means that organisms that don't show those resemblances or those shared features will be placed in another group. Those with shared features will be placed in the same group. Okay, so this has brought us to the end of the first part, the introduction. We've defined what taxonomy is, the science of classification. We've also defined what systematics is, simply grouping, assigning groups. When you assign groups to living organisms. Then we've also seen what you call classification, sorting out, or placing organisms into certain groups on the basis of their shared characteristics. Now, I'm going to the types of classification, but the main types of classification, are natural and artificial. So we have what we call artificial classification, and what we call natural classification. When we talk about artificial classification, we are mainly talking about, or we mean, placing organisms into groups on the basis of features without an evolutionary relationship. When we place organisms on the basis of shared features that do not have an evolutionary origin or evolutionary relationship, that is called artificial. Let me give you an example. We all know that insects have wings, and the birds also have wings. Wings are a feature in the insects, but at the same time, we find wings in birds. So if you place an insect and a bird in the same taxonomic grouping, then that is called artificial. Why? Because if we look keenly, if we examine the structure of a bird's wing and the structure of an insect's wing, although both are known as wings, they look to have a different structure origin, a different basic plan. For example, we know that that the wings are in birds. In birds, the wings are modified forelimbs. Birds were supposed to be tetrapods. Birds were supposed to walk on four legs. But what would be the front pair of limbs? The front pair of limbs was modified into wings that can enable the bird to fly. That can enable the bird to fly. Because most of them move in air. Therefore, they needed a device for flight. So, looking at the basic structure plan, the evolutionary origin of a bird's wing, it is actually similar to the hind limbs, which it uses for walking, because birds can walk on land using the hind limb, the hind pair of limbs on their 2 feet. They can. Look at your hands and any other birds. When they land on land, when a bird lands on land, it walks using the hind pair of limbs. But when it is flying in the air, that bird flies. We call it active flight, or flapping flight. That bird will fly with the front pair of wings, which it would use for walking. So compare that wing pair with the wing pair of insects. You will find that these are completely different origins, completely different basic plans. So you will find that the pair of wings in an insect is far different in structure origin or in structure plan from that of birds. So an artificial classification would place insects and birds in the same taxonomic grouping, simply on the basis of both having a pair of wings. So this means that artificial classification is not true natural classification. The opposite of artificial is natural. So if we base our classification on shared features that have a similar evolutionary origin or that have similar evolutionary relationship, then that is called a natural classification. Let me give you an example. Mammals, that class called the mammals, would include what we call placental mammals, and another group which we call non-placental mammals. So you will find placental mammals and non-placental mammals being placed in the same group because maybe both have mammary glands, or in both the body is covered with hair. So the hair, that feature called hair, and a feature called mammary glands is making us place a certain mammal and another mammal which may be living maybe in a desert environment, another one in a tropical forest, and another mammal such as a whale that lives in water. We place them in the same class, in the same grouping, mammalia, simply because we are using features that have similar evolutionary origin or similar basic plan. What does it mean? It means that natural classification shows what I call evolutionary relationships among organisms. Natural classification indicates evolutionary relationships among organisms in the same group. So if you find organisms in the same grouping, which grouping is based on natural features with the same basic plan, then we call that natural classification. So what does it mean? It means that artificial classification is not does not reflect the evolutionary relationships among the living organisms, because you can find me having placed a cockroach and a certain bird in the same group, and my reason will be both have wings, both have a pair of wings. So I'll place a housefly, and a hen in the same group, which is artificial grouping. And I will tell you, I've based on the fact that both have a pair of wings. So that is artificial. There is no evolutionary relationship. But if I use, for example, a structure, I can use a structure like this limb. This one is called my forelimb. This is a pair of forelimbs, and these legs are a pair of hind limbs. Now, if we are considering these limbs, when you go to a human has got what you call a pentadactyl. Penta means five digits. So there is one digit, the second, third, fourth, and fifth digits. So I have a limb, I have this limb with the 5 digits at the end. That is called the pentadactyl limb. But the same pentadactyl limb, you will find it in chicken or in poultry, or in some birds. You'll find for the birds, they may be having only 2 or 3 digits that you see, but there are also certain, the 4th and the 5th, these have degenerated, but they were there. So you'll find that in a bird, when you look at the legs of a bird or the wing of a bird, you are able to count the 1, 2, 3, 4, 5. Although there are only a few which are well developed, and others have degenerated. They have reduced in size. But the 5 digits are there. When you go to another group, which is not a mammal, maybe if you go to amphibians, these amphibians, you'll find them having webbed feet, the webbed feet. But when you try to count, you'll find that these digits in the hind limb of our limb are also 5. They are also 5. Although you may have like 3 or 4 which are well developed, others have reduced in size. They have degenerated. But there is an indication that their limbs were 5. We are having 5 digits, which we call pentadactyl limb. So if I use a pentadactyl limb to place Mr. Bukenya, you place a frog, you place chicken, some hen, and you place some reptile, such as a lizard. You place a lizard, you place me, you place a hen in the same group on the basis of pentadactyl limb. That both of us, I mean all of us, have got a limb which ends in 5 digits. Then that one is a natural classification. Why? Because it shows that Mr. Kenya, a frog, a lizard, and a hen might have originated from the same ancestor some millions of years ago. The hen, the reptile, the mammal or human, they were in the same group. That is why they are sharing, or they are all sharing that pentadactyl limb. So, such structures, we call them homologous structures. Homologous. Homologous, that word for homologous. Homologous structures. So natural classification is mainly based on homologous structures. The word homo means same. It means same. And an example which I've given you, which is the pentadactyl. Pentadactyl. The pentadactyl limb. That example which I've given you, which is called the pentadactyl limb, a limb with 5 digits. You will find it in reptiles, you find it in mammals, you'll find it in birds. I mean, diverse groups, but they show some similarity. And that is similarity, such as the pentadactyl limb is called a homologous structure. And it shows what we call common ancestry. It indicates common ancestry. Common ancestry means that those diverse groups, like the mammals, the birds, the reptiles, they share a common ancestor. They should have originated. They should have evolved from the same parents, from the same ancestor, millions of years ago. That is why the present day birds, the present day mammals, the present day reptiles are still showing some signs of similarity by having these homologous structures, although they appear to be very diverse groups, very different groups, but they show some similarity. So if we classify organisms on the basis of structures or features that reveal a common ancestry, structures or features that show evolutionary relationships, then that one is known as a natural classification. But if you place the water duck, the duck that lives in water, the water duck, the fish, and the whales. You see, these are three different groups. We all know that a whale is a mammal which lives in water. We know that the fish is another class of vertebrates. But we also know that a water duck is a bird. It is a bird. But you may find these three groups, maybe both of them having fins. If all of them have fins. Now, this characteristic, this feature of fins is not a homologous structure. It does not show common ancestry. For example, the fins in the whale, the fins in the fish, and those of the water duck, or the swim bladder in the whale and that in the fish might be basically different in structure. The basic structure may be different because we know a whale is a very large mammal, but it lives in water. A fish of different varieties also live in water. But when we say both have fins and then we place them in the same group, then that is called artificial classification. Artificial, why? Because the feature of fins that we have used to place the whale and the fish in the same taxonomic group, that feature is not, it does not reveal common ancestry. That feature does not reveal evolutionary relationship. So we call these features, we call them analogous structures. Analogous structures. Analogous structures. That word analogous structure means structures that perform the same functions in different groups of organisms, but which have different ancestry. Structures which perform the same function in different groups of organisms, but they do not have the same common ancestor. They do not have evolutionary relationship. I've given you examples: the wings in birds and the wings in insects. Those are analogous structures, and they are the ones which artificial classification uses to place two or more organisms in the same group. And therefore, they do not reveal evolutionary relationships among organisms in the same group. On the other hand, homologous structures, these are structures that may perform different functions, but have the same basic origin, or they have the same ancestry, the same ancestor. An example I've given you is the pentadactyl limb in the birds, pentadactyl limb in the reptiles, pentadactyl limb in the mammals, and in other groups of vertebrates. You will find in a bird, the pentadactyl limb may be used for flight. In humans, it may be used for walking or grasping or touching. In maybe the mole rat, the naked mole rat, it may be used for digging. The pentadactyl limb may be used for digging, or in the mouse. In the mouse, it may be used also for drilling holes. They drill their holes, and the rats, they use that pentadactyl limb. So in different groups of organisms, that pentadactyl limb is being used for different functions. But when we trace for the origin, the evolutionary origin of that pentadactyl limb, it shows that regardless of the group of animals where it exists, the pentadactyl limb shows common ancestry. And therefore, when we use homologous structures, we show what we call evolutionary relationships among organisms. The last class, or the last type of classification which I want us to talk about, is known as, now of course I said the two are the main, but modern day, the present advances in biology, the present advances in taxonomy have revealed that, or have shown that, we can use differences in DNA sequencing, that molecule known as DNA, which we call the hereditary material or genetically material, is universal in all living organisms. It is found in all living organisms. And DNA is made up of what we call nucleotides. The subunits of DNA are known as nucleotides. They are known as nucleotides. So if we have many nucleotides that are linked together, that are bonded or linked together, the resulting structure is a polymer. It's a polymer, a longer molecule, which we call a double strand or double helix of DNA. Now, this is natural, that each nucleotide is made up of three components. We have what we call a pentose sugar. Pentose sugar means 5-carbon sugar. There is what we call phosphate group. Phosphate group. And what we call a nitrogen base. Those three. A pentose sugar, which may be deoxyribose, a phosphate group, which is the same throughout DNA, and we have what we call a nitrogen base or organic base. Those three form one nucleotide. They form one nucleotide. But now, since we have the same sugar, which we call deoxyribose in DNA, the sugar is the same. It is called deoxyribose. We've called it a 5-carbon sugar. That means in the structure of this sugar, deoxyribose, there are 5 carbon atoms. And this is why I've called it a pentose sugar. Now, the second component in a nucleotide of DNA, I've called it a phosphate group. Sometimes it's called phosphoric acid, but we call it a phosphate group. We all know that a phosphate group is the PO4 3-. It is known as an anion, a negatively charged ion or radical. So these two, deoxyribose sugar and phosphate group, are always the same in all of the DNA, in all living organisms. In all living organisms, every nucleotide unit in DNA is made up of deoxyribose sugar, pentose sugar, and phosphate group. But what differs for each nucleotide is known as the nitrogen base or the organic base. So the third component of a nucleotide is known as the nitrogen base or the organic base. And it is this organic base, that brings that causes 4 different types of nucleotides that we can have in the DNA. Remember, I've said all of these three, they form what we call a nucleotide. And we are saying a nucleotide is the building block, the building units, the basic units of DNA. They are called. So when we have one nucleotide, another nucleotide, and we get that kind of double-stranded molecule, that polymer, we call it DNA. But I'm saying that the nucleotides are different depending on the nitrogen base they contain. For example, there are 4 nitrogen bases that we know in DNA. The first one is called cytosine, but we simply represent it with capital C. Another one is called guanine, which we can represent with capital G. Another one is called adenine, which we can represent with capital A, adenine. And another one is called thymine. Thymine. Those are familiar terms in cell biology. In cell biology, the nitrogen bases in DNA are abbreviated with the symbols C, G, A, and T. Now, what I'm trying to show you here is the modern branch of taxonomy, which we call molecular taxonomy. Molecular taxonomy. That modern biologists, modern taxonomists prefer classifying living things on the basis of the nucleotide sequence, on the basis of nucleotide sequence of their DNA. For example, let me speak this one before I write. If you have your father and you are the child, but there is also a grandfather, and there is also a great-grandfather. So those are 4 generations: great-grandfather, grandfather, father, and you. Those are 4 generations. In order to prove that you are a descendant, in order to prove that you are a descendant or a child of all of those parents in the 4 generations, we use DNA sequencing. DNA sequencing. That is to say, your DNA as a child should show similarity with the DNA sequence of your father, and that of your father should also show similarity to the DNA sequence of your grandfather. That of the grandfather should show a DNA sequence also similar to that of the great-grandfather. So, it is this molecular technique which is used now in molecular taxonomy to classify organisms. So they look at what we call DNA sequence or nucleotide sequence of your DNA. So basically they look at the order. The word sequence means order. The order in which the nucleotides with these bases occurs within living organisms that we want to classify. Cytosine is a nitrogen base, but it is different from guanine. Guanine is also different in appearance or structure from adenine. Adenine is different from thymine. But these 4 bases, organic bases, are the ones that make the different units, the different nucleotides in DNA different from one organism to another organism. So the nucleotide sequence in the DNA of one organism is always different from the nucleotide sequence in the DNA of another organism. So when we use the nucleotide sequence in DNA to classify living organisms, as it is done in molecular taxonomy or in modern taxonomy, that type of classification is known as phylogenetic classification. We call it phylogenetic classification. It is based on DNA sequence. It is not very easy because in Uganda, for example, we have very few molecular taxonomists. Those who can examine the DNA sequence and then classify living organisms. I would like to end by reminding you the reasons why we classify living organisms. Why do we classify living organisms? I will simply write why classify. Why classify living things? Now, one reason for classifying living organisms, which we have already seen, is to show or to reveal evolutionary relationships among organisms. To reveal evolutionary relationships among organisms. So when we have organisms placed in the same group like mammals, organisms in the same group like a fish, it shows that all the members in that group share some common ancestry. They might have originated from the same ancestor some thousands, some millions of years ago. So the first reason is to reveal evolutionary relationships among organisms in the same group. To reveal evolutionary relationships among organisms in the same grouping. But I don't want you to forget that this one is the goal of natural classification, which we have defined as the classification based on homologous structures. Classification based on structures that have similar basic origin. That one is one goal of classification. The most obvious goal, the most obvious reason for classifying is to create order out of chaos. Simply that. To create order, to avoid the confusion, to create order. And avoid confusion or chaos. One thing which we forgot to talk about, but which I want to point out now, the two main branches of taxonomy are the systematics, but the second one, which I call the classification, I want us to make some change. We call it nomenclature. Nomenclature. This one simply means the science of naming organisms. The science of naming organisms is called nomenclature. That is the second branch of taxonomy. So I want us to correct something small. That two branches of taxonomy. One involves grouping organisms, and we call that systematics. Now, the second one is called nomenclature, which is naming. It simply means naming organisms. Naming organisms. So, when we name organisms, then we avoid the confusion. For example, suppose there were 10 boys in a class, but all of them do not have a name. They don't have names. So you would just, and the teacher is in class. How would the teacher refer to them without a name? He would say, you boy, you boy, you boy, come. You boy, come in front. And this teacher would struggle to say, you, by pointing to the specific boy he needs. He would say, you, you come. And then many people will say, me, the other one, the other one. So a name is very important in biological classification. A name is important. That after we've placed organisms in the same group, then we assign a name to that group. Or we assign a name to that living organism. For example, in the whole world, human beings are called Homo sapiens. Homo sapiens. That is the name for humans. So we are different from gorillas. That name makes us different from chimpanzees, although we show some resemblance with those animals. But for us, we are Homo sapiens. Sapiens means wisdom. Wisdom. We are intelligent. We show some higher degree of intelligence. We can build houses. We can cultivate. We can cook. We are no longer living in forests. We are no longer doing things in the open. We have houses, homes, but our relatives, the closest relatives of human beings, are called the chimpanzees. Our relatives are still where? In the forest. And they are fruit eaters. They mainly eat fruits. They live in maybe caves or in trees. So Homo sapiens is a unique group made up of all intelligent humans. Other primates that do not have a well-developed brain like that we have are placed down in another group. So whenever we are classifying, we avoid confusion by assigning specific names to different groups of organisms, to different groupings of organisms. Now, that thing has also brought me to what I call the diversity of living organisms. There are millions of plants, millions of animal species, millions of other species of bacteria and so on, some of which are known, others have not yet been discovered. And the world would be full of chaos or confusion if these things, if the biodiversity, the biological diversity is not classified. There would be a lot of confusion between the plants we know and those that we don't know. But for those that we know, those which we have discovered, we've given them names. And those which we are yet to discover, we will study their features and also assign them groups and names to which they belong. So, classification brings order in a complex world of living things, in a complex world of very many living things. And the third reason for classification is what I've been explaining. I gave you an example of the name Homo sapiens. That is a Latin name, Homo sapiens, for humans. Humans are called Homo sapiens, the scientific name, which is actually a Latin name, because science began in mainly Latin and Greek languages, not in English. So the scientists agreed that the Latin names will be retained, they will be maintained when referring to names of living organisms. Remember, the botanist who was called Carl Linnaeus is the one who introduced this system of naming, where every organism is assigned a name that contains two names, the generic name and the species name. The generic name shows the genus to which the organism belongs. The specific name or species name shows the species to which the organism belongs. So a human being belongs to a genus, Homo, and a species, Sapiens. So we combine the two names to form the scientific name, the Latin name of this organism, of this species, and we call it Homo sapiens. So the third reason for classification is to assign a universally accepted name, a universally acceptable name for use by all biologists in the whole world. So to assign an internationally or globally recognized name. Recognized name, which we are calling the scientific name. I've already told you that if you are in a class and you have 10 pupils or 10 students without names, you will struggle to get exactly to the person you need, because you'll just say you, because they don't have a name. So living organisms should be given names that are universally or internationally recognized, whereby if somebody is in Arab world and says Homo sapiens, we all know is referring to humans. Somebody may be in Japan, another one may be in America, another one may be in Africa, another one may be in China. All will use the word, the name Homo sapiens to refer to a human in any scientific conference, in any scientific meeting. Only one name, universally, internationally recognized by all scientists. So that is another goal for classification. But also, we know that we also know that there are very many living organisms that have not yet been identified, as yet. And now, there are some groups, there are some taxonomic groups that we know already. For example, we know birds, we know mammals, we know reptiles. And if a new organism is discovered, whose resemblance, for example, is like that of a bird, and on the other hand, it's like a reptile, and its identity is not yet known, taxonomists can place that organism in a certain grouping depending on the features that it shows or that it has, which are similar to the already known organisms. So we can easily identify a new organism on the basis of what we already know by examining or observing the features that it has. So the fourth reason is simply is the identification. Is the identification of organisms. Remember, we have got very many plant and animals, plant and animal genetic resources out there in the world that have not yet been identified. But their identification will depend on a comparison of how they look like with the representative organisms in taxonomic groupings that we already know. And when we do that, we will likely place that new organism in a certain taxon, in a certain taxonomic grouping, maybe in some class, or in some order, or in some genus, because a variety of organisms there are not yet named, they have not yet been identified. Those which have been identified are fewer than those not yet identified. So taxonomy or classification enables easy identification of organisms. But like I told you about diversity of organisms, diversity of plants, diversity of animals, classification enables us to learn or to understand plant and animal diversity and diversity of other organisms that exist on Earth. We understand and appreciate. We understand and even appreciate the diverse forms of life, the diversity of organisms, or diversity of the life forms that we have on Earth. The diversity of life forms that we have on Earth. Now, we are talking about reasons for classifying living organisms. I've said classification enables us to get an internationally recognized name, which is the Latin name or scientific name. It allows us to appreciate the diversity of organisms on this planet. It allows us to create order out of chaos, And other reasons I've given you. But you should never forget that classification also is important in research. Research simply means that they are ongoing studies. They are ongoing inquiries about what we don't know, about what we do not yet know. Probably that research could be of medical importance. The research could be of agricultural importance. Or it could be of maybe veterinary importance. For example, there are many undomesticated plants out there in the wilderness whose characteristics we do not yet know, yet they are edible. They are edible plants, they are edible fruits, they are edible things out there for animals, for our livestock, for humans. But we have not yet done enough research. So, if this work of classification goes on, and we understand the characteristics, the features of many undiscovered, unnamed plants out there, unnamed animals out there. We can understand their genetic characteristics and how we could maybe exploit those characteristics to improve the present day varieties of crops, the present day varieties of livestock that we have, the present day variety of poultry that we have. They are there. For example, if you do some genetic cross between some poultry in some grassland or forest and the poultry that we have in our homes, you could get maybe a fat growing chicken. You could get maybe some chicken that lays a very big egg that you can even sell at 2,000. But enough research has not yet been done to understand the characteristics of organisms out there in the world, in the wild. So we are saying another reason for classifying is for scientific research, for scientific research and scientific innovations. Scientific innovations. And maybe last but not the least important, as I've already informed you, is that classification could improve agriculture, it could improve medicine, it could improve domestic livestock and poultry productivity. So we should not play around with this science of classification. In another video, I'm going to share with you what we call linear classification, what we call binomial system of classification that I've already told you about. That a human is called Homo sapiens. It is given two names, a generic name and a species name. That is called binomial nomenclature or linear classification. And it will be our next focus in the coming video. But also, we will also look at the diversity of organisms. We will introduce what we call biological diversity. And we are going to see that there are different levels of biological diversity. We have what we call genetic diversity. Then we have what we call species diversity and ecosystem diversity. So biological diversity could be looked at three levels: genetic diversity, species diversity, and ecosystem diversity. So we will look at that next time. Also, in the same course, we will look at what we call taxonomic hierarchy or taxonomic grouping, where we look at the 7 kingdom classification. Not 7 kingdom, 7 taxa, 7 units of classification. We will begin with the kingdom, followed by phylum, followed by class, followed by order, followed by a family, followed by a genus and a species. That is called the 7 unit hierarchy, the 7 taxa hierarchy, taxonomic grouping. If we are grouping, if we are taxonomically classifying a human being, we will begin with the kingdom to which the human belongs, which is kingdom Animalia. Then we go to the phylum. to which a human belongs, which is phylum Chordata, which is the phylum Chordata. Then we shall talk about the class to which a human belongs, which is called class Mammalia. Class Mammalia. Then we shall talk about the order to which a human belongs, and that is called the order Primate. The order Primate. Then we shall talk about the family to which a human belongs, and that is called the family Hominidae. Hominidae. It ends with DAE, DAE, Hominidae. Then we shall look at the genus to which a human belongs, and that is Homo. Homo. And then the species to which a human belongs, and that species is called sapiens. Sapiens. So that is called 7-unit hierarchy of classifying an organism fully. We will look at it in detail in the next video. In the next video, we'll also look at that. I've already talked about the domains of life, but I will not repeat them. We've already covered them. Domain eubacteria or prokaryota, domain archaebacteria, and domain eukaryota or eukarya. And that one we shall not look at. Then we'll also look at the 5-kingdom classification. 5-kingdom classification, which begins with the bacteria kingdom, bacteria. Another kingdom is kingdom protista. Another kingdom is kingdom fungi. Another kingdom is kingdom plantae. You end with AE, plantae. And another kingdom is kingdom animalia. That will be the business of the next video. Then we will explore the diversity of animals and plants, especially the higher forms of plants and animals. Animal diversity will focus on the lower the lower animals, what I can call the invertebrates. Those animals without a vertebral column, those animals without a backbone, and they do not have what we call a notochord. Notochord. The notochord is absent in these animals. They are mainly called invertebrates. So these are divided into groupings, large groupings, which we call phylum, large phyla. So each of these groups of animals is called a phylum, L-U-M, a phylum. And they are all invertebrates, but they're divided into larger groupings. For example, the segmented worms, such as the earthworm, Lumbricus terrestris, the earthworm, belongs to a phylum known as Annelida. So we call them the annelids. So the leeches, the earthworms are under this. Those earthworms which you see when it has rained, walking or moving, not walking, but moving around, they are called annelids. Another phylum which we'll look at is called mollusca. These are soft-bodied invertebrates, soft-bodied animals like the snails. You see them with the shell. You see them with generally with the soft bodies and living in damp environments, moist environments. They are mollusks. Phylum mollusca. Now, the largest The largest phylum in the animal kingdom is called Arthropoda. Arthropoda. A-R-T-H. Phylum Arthropoda. Now, this is the largest phylum in the animal kingdom. But also, we can say it is the largest phylum among the invertebrates. And these are many. When you talk about insects, that is phylum Arthropoda. When you talk about the arachnids, such as the ticks, the mites of Roro, the ticks, the mites, the spider, all of those are called arachnids. Arachnids. With the 4 pairs of legs and only 2 body parts, 2 main body parts. They have a cephalothorax and a head, like you see the spider and the tick. Those are arachnids, and they are a class in the phylum Arthropoda. When we talk about the diplopoda, such as the millipede, a gongolo. That millipede, gongolo, is diplopoda, class diplopoda. It is also an arthropod. And when we talk about the chiropoda, such as the centipede, the cigar, chiropoda, okamwaka, chiropoda, such as the centipede, you are still talking about a class in phylum Arthropoda. When you talk about the crustaceans, when you talk about the crustaceans, mainly aquatic invertebrates. That class, crustacea, is also in the phylum Arthropoda. So it is a very large phylum in the animal kingdom with those 5 classes. Class Chiropoda, class Diplopoda, class Insecta, class Arachnida, and Crustacea. So that one will be the business of the next discussion. Cnidaria. That phylum called Cnidaria. Okay, we don't read it as Cnidaria, but we kill the C and we begin with N. So we read it as phylum Cnidaria. Not Cnidaria, but Cnidaria. Like when you're writing psychology, you kill the P and you begin with the S. So even here, you kill the C and you read it as Cnidaria. Phylum Cnidaria covers simple forms of life, mainly found in marine waters or marine environments, like on the coast of an ocean. And you will find them bound, they are bound to a hard surface, such as what we call polyp, which is known as a coral. The organism is called a coral. An example is this polyp, but they are found mainly at the coast of the oceans in marine environments. When you talk about the sponge, most of these organisms are not mobile. They may not locomote from one place to another. They may be attached to a surface. But when you examine their cells, they have true cell membranes of animal cells. They also show some systems, like a digestive system, like that of an animal. They show some defense system, like that of an animal, and even the way they capture their food, the way they capture the prey. using what we call tentacles and nematocytes or nematoblasts, stinging cells to attack and kill the prey. They show that they are true animals, although lower forms of animals, lower forms of animal life. They are there in the coast, the sponge, the hydra, which we know reproduces mainly asexual by budding, that hydra. And these simple forms of life, the spongy, the hydra, the corals, and the phylum Nidaria, we mainly know they have a body wall that is made up of two layers, two cell layers. The outer cell layer is called the ectoderm, and the inner cell layer is called the endoderm. So they mainly have simple tissues, But in these forms of life, the lower forms of life, complex, we don't find their organisms. We also do not find their systems. You don't find their systems, but they have tissues, specialized cells carrying out food capture, specialized cells carrying out defense, specialized cells carrying out like digestion. But there are no systems, there are no organs. They are simply simple forms of life. Phylum Nidaria and invertebrates. And when we come to the group of vertebrates, which we say vertebrates are a subphylum under phylum Chordata. It is not a phylum. Vertebrates is not a phylum, but we call it a subphylum. The main phylum is the one which I called phylum Chordata. So all of vertebrates belong to phylum Chordata due to the presence of a notochord. A notochord is the precursor. It is that structure that leads to the spinal cord and the brain. The structure which leads to a vertebral column of backbone and the brain covered in the brain case or the cranium. So that notochord has been shown to be present in all the fishes, in all the amphibians. These are now the classes of vertebrates. In all, in reptilia, class reptilia, in all members of class the apes or the birds, and in all members of the class Mammalia. So those are the five classes: the Pisces, class Amphibia, class Reptilia, class apes, and class Mammalia are all classes of vertebrates. And I've said vertebrata is a subphylum under phylum Chordata. Under phylum Chordata. We are ending with what we call plant diversity. And we have grouped plants into two major groups: the lower plants and the higher plants. Remember, the main difference between the animals and plants is the type of nutrition. The plants are able to make their own food by photosynthesis. And we call that type of nutrition autotrophic nutrition. Autotrophic. So this one is show auto meaning same food. Autotrophic nutrition. That is one fundamental difference between plants and animals. Now, for animals here, They are unable to make their own food. They feed they obtain nutrients, animals obtain nutrients from other organisms. And we call that heterotrophic nutrition. So the major difference is the type of nutrition. Animals, heterotrophic nutrition, and plants, autotrophic nutrition. Another main difference, animals are able to carry out locomotion. They are motile, moving from one place to another. But the plants are non-motile. They are non-motile. What moves are the seeds, the spores, and other propagules or reproductive structures. But these animals carry out locomotion. No locomotion here. Plants have cells that are made up of cell walls composed of cellulose. They are cell walls in plant cells composed of cellulose. The cells of animals lack cell walls. They are not cell walls in animal cells. But here they are cell walls made of cellulose. So what I'm trying to show you are basic differences. But you should know that when we talk about plant diversity, then we have moved away from the conversation on animals, and we are now on a conversation of plants, which are autotrophic, which are non-motile, and which have cells with cell walls. So we are grouping plants into two groups, the lower plants and the higher plants. Now, whenever you hear about lower plants, what should come in your mind are lower forms of plant life, lower forms of plants. And the first lower form of plants, which we mainly find in damp environments, they grow on walls which are damp or watery. They are found everywhere, wherever it is damp or moist. They call them the bryophytes, such as the mosses. The mosses. And why are we calling these two, the bryophytes and the pteridophytes? This one is read as pteridophytes. So we kill the P, and we begin with the T. We read it as pteridophytes. The bryophytes and pteridophytes are being called lower plant forms. It is because they lack these lower plants. They lack the tissues, such as the vesicular tissues, phloem and xylem, that we find in higher plants. They don't have a well-developed vesicular system made up of phloem and xylem. No vesicular tissues. No phloem. Let me write here. There is no phloem. PH. No phloem. No xylem. Now, whether we are talking about a root, a stem, or a leaf, the three main parts of a plant, root, stem, or leaf, in higher plants, such as the trees and the herbaceous plants, we find there a well-developed root and shoot system. In the leaves, in the stems, and in the roots, you'll find there a phloem. This is for conducting the organic substances, organic solids, conducting the products of photosynthesis from where they are made to where they are used. That is the major function of the phloem. We call it translocation. Translocation. And that word vesicular means that the components are like vessels. They are like tubes, like this. So the food or water moves in tubular structures, such as the xylem vessels or the phloem sieve tubes. Now, those are absent in lower plants. We don't have the phloem for translocation of the organic products of photosynthesis. We don't have xylem for transporting water from soil to the leaves. We don't have the xylem in the roots, no xylem in the stem, no xylem in the leaves of lower plants. What they have, what these lower plants have are root-like structures. Root-like structures that are known as the rhizoids. Root-like structures known as the rhizoids are for anchoring, anchoring for anchoring the plant into soil or into the substratum or wherever it is found. You will not find there a true root. They are not true roots. A true root has a vesicular band made up of xylem and phloem, maybe in the center or somewhere, but it also has what you call a pith in the center. It has a cortex. The true root has a cortex. It also has an epidermis that is absent in the mosses and in the ferns, or what you call the pteridophytes. They are not there. So we will also see what we call alternation of generations. These plants, the lower plant forms, like the mosses and the ferns. The mosses and the ferns, they show what you call alternation of generations. The word alternation of generations means that in the life cycle of mosses or of the ferns, there are two prominent structures. We have two prominent structures in the life cycle. We have what we call a gametophyte. A gametophyte. During a certain part of the life cycle, this gametophyte, which we also call the gamete producing organ, the gamete producing organ, you will find it well grown, well developed, prominent in a certain part of the life cycle. In another part of the life cycle, you will find another organ or structure known as a sporophyte. Sporophyte is a spore producing organ. A spore producing organ. These two alternate in the life cycle. The gametophyte alternates with the sporophyte in the life cycle, and that is called alternation of generations. So it is a phenomenon in the life cycle of bryophytes or pteridophytes, where there are two distinct stages, two distinct organisms, a gametophyte and a sporophyte, alternating with one another in the life cycle. And that is the lower plants, whether it is the mosses, which are called bryophytes. The phylum is called bryophyta, and the plants are called bryophytes. Another example is called the liverworts. The liverworts. So the mosses and the liverworts both belong to this phylum Bryophyta. They both belong to phylum Bryophyta, mosses and liverworts. What have I said? I said they don't have true roots, no true leaves, no true stems. Actually, the leaves in these ferns, for example, the pteridophytes, an example is a plant known as a fern. The fern plant, F-E-R-N, a fern, belongs to phylum Pteridophyta. A fern. But I've said they don't have true roots, no true leaves, and no true stems. They instead have rhizoids in the place of roots. In the place of roots, they have what you call rhizoids. When it comes to the leaves in the fern, the cluster of leaves in a fern, we call it the frond. The frond. So they are not true leaves, but they have these fronds. And I cannot say that they are photosynthetic. I cannot conveniently say that they are photosynthetic. That means that if they can photosynthesize, the powers are very low compared to the higher plant forms of life. So those are the lower plants. I don't want you to forget the following points. Number one, the two main groups of lower plants are the pteridophytes, like the ferns, and bryophytes, like the mosses and the liverworts. But both of those groups don't have vesicular system of phloem and xylem. They are absent. And probably that is the reason why their size is simple. Their size is simple because they don't have conducting tissues for water and food. Second, I've said we don't have a true root system and no true root shoot system. So we don't have true stems and true leaves for the shoot system. They are not there. What do they have? They have rhizoids for anchoring in soil and some leaf-like structures which we call frond that we find in the ferns or in the pteridophytes. But the real vesicular system is absent, and that is why they are simple or lower forms of plant life. Then when we go to the higher forms of plant life, we have what we call the spermatophytes. And here I've written that spermatophytes are seed-bearing plants. All seed-bearing plants, the plants which have seeds, the plants which produce seed, seed-bearing plants are known as spermatophytes. They are known as spermatophytes. And we have grouped these spermatocytes, we've grouped seed-bearing plants into two main groups, two main phyla. Now, in botany, when we are discussing plants, instead of using the word phylum, we prefer to use the word division. In the place of phylum, phylum is for zoology, animal taxonomy, animal classification. We can use phylum. But when we come to plant classification, we prefer to use division. So there are two main divisions under this group of spermatophytes. The first division is gymnosperms or gymnospermatophyta. And another division is angiosperms or angiospermatophyta. Angiospermatophyta. So the gymnosperms and angiosperms are both seed-bearing plants. The main difference is that gymnosperms are non-flowering plants, and since they don't have flowers, the seeds are produced in what we call cones, in the conical structures, which are called cones. The seeds are formed in cones and not from the ovaries or fruits, but what they have are called cones, and their seeds are called naked seeds. They are naked seeds because gymno means naked, and the other sperma is seed. So naked seeds. So gymnosperms, such as oroconifers, when you find like the pine, when you find like fir, F-U-R, when you find redwood, when you find cypress and the other cone-bearing plants, whose leaves, these trees, the coniferous trees, they have leaves which are needle-like or scale-like. The leaves are pointed, and we mainly find them in some American, North American, and South American parts. The coniferous plants, they are a group of gymnosperms. Other groups of gymnosperms apart from conifers are called cycads, like the cycads. Okay, but we are interested in the main group. The main group is the conifers. Cone-bearing plants, naked seed plants. So here, in the conifers, you also find some form of pollination. But the pollination here is aided by air currents or winds. Air currents or winds moving pollen grains from the male part of the cone to the female part, in order to bring about fertilization, resulting into formation of a seed. When we come to angiosperms, but before we talk of angiosperms, the redwood, which is the tallest tree known on earth, the tallest, over 100 meters tall, is a coniferous tree or a coniferous plant. So you will find these plants mainly growing very tall, but with a smaller girth, smaller cross-sectional area of the stem. But when it comes to height, they grow very tall. The second group of higher plants is angiosperms or angiospermatophyta. These are the flowering plants. So whenever you hear flowering plants, then what should come in your mind is that word, angiosperm or angiospermatophyta. In other words, the reproductive structure in this group are the flowers. Which flowers bear the female part, known as episteal or gynoecium, composed of a stigma, a style, and the ovary with the ovules. Then the male part that we call stamen or androecium, a whole of stamens, whole, WHORL, a whole of those stamens. It is called androecium. Androecium. So those are the two terms that you should remember. When we talk about androecium, we are actually talking about the stamens of the male parts of a flower. And when we talk about the gynoecium or gynoecium, we are mainly talking about the pistil of the female part of a flower. So the androecium or the stamens bear the anthers and the filament, but the pollen grains that contain the male gamete are contained in the anthers. And when they are mature, they will split open and release these pollen grains, which can be carried either by wind or by insects to the gynoecium, to the pistil. And when those pollen grains land on a mature stigma, they will germinate and form a pollen tube that grows down the style to the ovary. Then it enters through a small hole known as a micropyle. But on its way to the ovary, the nucleus in the pollen grain will divide mitotically to form a tube nucleus and a generative nucleus. Okay, the tube nucleus leads the way and the generative nucleus falls behind. It trails. Eventually, double fertilization takes place. So, angiosperms show double fertilization, resulting in formation of an embryo, which becomes the seed, and endosperm, and a primary endosperm tissue is formed also as a result of fertilization, which provides food or nourishment for the growing embryo. So there is double fertilization in angiosperms, which we did not see in gymnosperms. But interesting for you to know is that there are two classes of angiosperms which are known. The monocotyledons, or class monocotyledoni, and class dicotyledoni. This simply means plants that produce seeds with one cotyledon, such as the maize or the corn, wheat. All the grasses are monocots. You will find there plants like the palm, they are monocots. Huh? The palm, okay, mainly the grasses. Let us say mainly the grasses. But what we know about these monocots, when you look at the venation in the leaves, they have parallel venation. They have parallel veins or parallel venation. The leaves of the monocots are these, like in maize plant, you'll find parallel venation. But when we come to the dicots, the leaf, you will find a leaf showing what we call, if this is the midrib, then it shows net venation or reticulate venation. Reticulate, a network of veins spreading throughout the entire leaf and transporting. These veins contain xylem for distributing water in all parts of the plant, in all parts of the leaf, mainly to the chloroplasts in the plant cells to enable photosynthesis to take place, because water must be distributed to all plant cells because they are photosynthetic, and water is a raw material for photosynthesis. So the network of veins or the parallel veins all enable, they contain xylem, which distributes water from soil to the different parts of the leaf to carry out photosynthesis. The other difference is that dicots are made up of two seeds. They have two seeds. They have seeds with two cotyledons. Dicots have seeds with the two cotyledons. Another name for cotyledon is seed leaf. Seed leaf. When a plant is germinating, when a seed is germinating, the first seed leaves that are formed, the first seed leaves that are formed could be one in monocots, if it is, for example, a maize seed, or it could be two seed leaves that are formed if it is a bean seed. So the beans, most of the trees, the peas, all of those are dicotyledonous. But what is also important is for you to note the root system. The dicots mainly have a taproot system, a taproot, which anchors that plant, that tree deeply into soil. But when you look at the root system of monocots, they are mainly fibrous roots. They are fibrous roots. So the fundamental difference is that the tap root is mainly strong, and it anchors the plant into soil deeply. Then the fibrous root system, it mainly spreads. But this is not the adventitious. The fibrous, they are like fibers that are spread. Like the root system of grasses go out and uproot any grass. That root system you see is a fibrous root system, and it is a characteristic of monocots. Then the taproot system is a characteristic of dicots. So I ask you, ladies and gentlemen, to go and make more and more reading on these components of our course. But concerning our assessment, what I have covered in today's video is enough. Just review that video from the start to the end, and you'll find the examination, the assessment, very cheap for you, because all questions will be straightforward, requiring the same knowledge I've shared with you briefly in this video. I wish you good luck, and God willing, after the COVID time, we'll be able to meet face to face, and we continue with the conversation on different secrets and aspects of biology. God bless you. I wish you good luck as we build for the future, for God and my country. Thank you.